Welcome to Physiology for Students. I'm Dr. S. Today, we're going to talk about genetics. This clinical physiology lecture is the third in our series. It's genetics and protein synthesis. There are two parts to today's lecture. I'm breaking this up because it's a bit of a long lecture, and I want to make sure you can go through all of these very important details. We're going to spend some time looking at DNA, the structure of DNA, and some basic concepts related to genes and gene expression. We'll talk about the process of transcription and translation. And then, in part 2, after a little bit of a coffee break, we're going to talk about how genes are regulated and how gene expression is regulated. Both parts of this lecture are very important, especially as we think about the importance of genetic disease and gene alterations that can alter the function of a cell. Genetics is a branch of biology concerned with genes, genetic variation, and heredity. Our genes make us who we are. Genes are composed of segments of DNA found within the nucleus of the cell. Genes control cellular functions through the proteins that are produced by the cell. Genes also determine heredity and what traits or characteristics are passed on from parent to child. Heredity is the passage of traits from parent to a child, one generation to the next. A trait is a physical or mental characteristic. I always talk about the redhead trait in my family. I have 2 beautiful redhead children. Of course, their mama is a redhead. How is that possible? Well, I passed on the gene that gave them their red hair. We all know that we resemble our parents and our family members with our physical characteristics. But there are also microscopic characteristics in our cells that change the way our cells function. Some of that is inherited. Some of that is due to our life experience. It's important to look at the difference between genes within somatic cells and genes within reproductive cells. Somatic cells are all body cells. As we're exposed to mutagens, or things in our environment that can change our DNA, that can alter the DNA within our somatic cells during our lifetime, for example, UV light exposure. But these alterations will not be inherited. So you may have, for example, excessive UV light exposure to an area of the skin that causes a genetic mutation and ultimately a cancerous skin tumor. That's not going to be passed on to your children. That's simply in that group of cells. Reproductive alterations, however, within the gametes, which in females would be the oocytes or eggs, and in males would be the spermatogonia or sperm, those changes can be inherited and passed on to your offspring. So only changes within reproductive cells are passed on to offspring. Sometimes this is a good thing. It causes genetic variation. And if you think about the theories of evolution and natural selection, it's these tiny changes in genetic variation that allow the changes that can help an organism thrive and survive. Some of these changes are not good and can lead to mutations that can alter the function of the cell and ultimately create a loss of function or even be fatal. So now that we've got that out of the way, let's look at some basic terms related to genetics. Gene expression is the process of making proteins from the genetic code. The genetic code starts with the DNA in the nucleus. DNA, however, is protected and cannot leave the nucleus. So there's an intermediate that reads the DNA and sends that DNA code into the cytoplasm of the cell. That's the mRNA. The mRNA is the messenger RNA that goes out to the cytoplasm of the cell to direct protein synthesis or the formation of proteins within the cell. Remember that proteins have so many functions within the cell. Structural proteins, signaling proteins, enzyme proteins. So this really is the basis of the function of the cell. Let's briefly review the structure of DNA. DNA, deoxyribonucleic acid, is composed of nucleotides. Each nucleotide has 3 basic chemical groups: the phosphate, the deoxyribose sugar, which is where the D in DNA comes from, and one of 4 possible nitrogenous bases. These nitrogenous bases are adenine, thymine, guanine, or cytosine, which we shorten to A, G, T, or C. The phosphate groups and the sugar groups are circled here. And then the bases, or the nitrogenous bases, are each in these 4 possible DNA nucleotides, A, G, T, or C. These nucleotides form the letters of the DNA genetic code. So a single strand of DNA is formed first with a sugar phosphate backbone, and that is the phosphates and the deoxyribose sugars line up on the outside of the DNA strand, forming a backbone of sugar phosphate groups bonded together. And then pointing inward, we have the nitrogenous bases. Here we have a T and a G lining up next to their sugar phosphate backbones. The final double-stranded DNA molecule is formed when a mirror or complementary strand matches base pairs at the center with its sugar phosphate backbone on the opposite side. So let's look at that structure here. So here on the left side, we have the sugar phosphate backbone. You can see phosphate, sugar, and the nucleotides T and G accompanying those phosphate sugars. And then on the other side, we have on the very outer edge, again, the sugar phosphate backbone. There's phosphate, sugar. And you'll notice that on the other side, the base pairs are pointing inward. On this side, we have an A and a C. The base pairs bond together in a very specific set of complementary base pairs. A and T always go together, and G and C always go together, just as shown in this diagram. So you'll notice that the thymine and the adenine are bonded together, and the cytosine and the guanine are bonded together. And this forms double-stranded DNA with a sugar phosphate backbone on either side and the nucleotides pointed inward, bonding together with these base pairs, A's to T's or T's to A's, G's to C's or C's to G. There's a lot of chemistry about why the DNA double strand forms a double helix. That's not actually extremely clinically relevant for us. We want to move on a little bit more to talking about how the DNA genetic code is used. But if you're interested, you will find resources that talk about the double helix, how it was discovered, and all of the science behind that double helix. But let me show it to you here. So here you can see that as the sugar phosphate backbone wraps around on either side, it forms a double helix. A helix is a twist, a double twisted shape. So we have 2 strands of DNA that twist to form double twists or a double helix. One of the sugar phosphate backbones goes in a 3' to 5' direction, and one of the sugar phosphate backbones goes in a 5', my arm doesn't go that way, 5' to 3' direction. So they're exactly opposite each other. We'll talk about this in a moment. So let's review DNA structure. It might be helpful for you to try to draw this out. So look at the phosphate group, the sugar group, and the nitrogenous base. Look at the overall DNA structure and see if you can find those groups bonding together to form that double-stranded complementary base pairs that ultimately will twist to form that double helix. And remember, the letters are A, T, G, and C. A's always bond to T's, T's always bond to A's, G's always bond to C's, C's always bond to G's. Keep that in your memory. It's very important. Within the nucleus, these long strands of DNA code are organized into chromosomes. Chromosomes are long sequences of DNA wrapped in histone proteins. There are 23 chromosomes in the human nucleus. 22 of those are autosomal, numbers 1 through 22, and the last chromosome is a sex chromosome determining male or female sex, having an X or a Y chromosome. So every nucleus has 23 types of chromosomes. We get one set of 23 chromosomes from our mother and one set of 23 chromosomes from our father. So we have 23 pairs of chromosomes, numbers 1 through 22 autosomal, and the X or Y. Within these chromosomes, there are long sequences of DNA, each sequence being a gene or a non-coding region. The coding regions of DNA, or the genes, form a code based on triplets of nucleotides. So this is the triplet code. For example, we'll break this up into ACC with its complementary TGG. That's one triplet there. Each triplet ultimately encodes an amino acid that will be one amino acid in a long stretch of protein. Remember that amino acids bind together to make peptides. Many amino acids together make a polypeptide and then eventually a protein. So in this triplet code are the letters that form the code for what will ultimately be an amino acid in a protein chain. There's a template strand and a coding strand of the DNA. The template strand has its sugar phosphate backbone in the 3' to 5' direction. The coding strand, or the complementary strand to the template strand, is in the 3' to 5', is in the 5' to 3' direction. So the coding and the template strand have their sugar phosphate backbones in the opposite direction. Okay, I should have done that in the other way. So 3' to 5', 5' to 3'. The 3' to 5' forms the template strand. Why do we care? Because eventually we're only going to read one strand. And guess which one we read? We read the template strand. So the importance of understanding the sequences of DNA is to be able to figure out what they encode and how it will be read. DNA has to stay in the nucleus, however. So for the DNA to create a protein in the cytoplasm of the cell, there needs to be an intermediate. This is where RNA comes in. So RNA is used to read the DNA and take that DNA message, sort of like a recipe, out to the cytoplasm of the cell, where then the final protein can be made. So I think of the DNA like a genetic library of recipes, super top secret recipes that can't leave the nucleus. And the RNA is like when you go to your grandma's house and you write down that recipe and you bring it back to your house so you can make it hopefully as good as grandma's, right? So the RNA reads the DNA sequence, creates its own message, the messenger RNA, and then travels to the cytoplasm where the protein can be made from that mRNA message. mRNA codons then are also in the form of 3 bases because they're simply reading the triplet code of the DNA, but they're going to be complementary. So let's take a look. Here's our template strand of DNA running in the 3' to 5' direction. We're going to use that to bring in our RNA and read the code and copy that code. RNA then comes in and forms nucleotides that are complementary or reading exactly that DNA code. And they're going to run in the 5' to 3' direction, just like the coding strand of DNA. Why? Because they're both complementary to the template strand. One thing you'll notice is that the RNA structure is very different than the DNA structure. So let's take a moment to review the structure of RNA. So here we're talking about mRNA. It's single-stranded and formed of nucleotides, but slightly different nucleotides than the DNA nucleotides. Phosphate, same, but a different sugar, ribose sugar, which is where RNA gets its R. So DNA has deoxyribose sugar, which is where it gets its D, and RNA has ribose sugar, which is where it gets its R. And one of the 4 possible RNA bases. The big difference between DNA and RNA is that there is no T nitrogenous base. There is only a U. So the bases for RNA are not ATGC, they're AUGC. And A's always bond to U's and U's always bond to A's. So here we have adenine, uracil, guanine, or cytosine possibilities for the RNA bases. And those are pictured here: cytosine, guanine, adenine, and uracil. This is a nice diagram for comparing the DNA structure to the mRNA structure. So let's take a look. Here we have the double helix of DNA and its complementary base pairs in some sequence of A, T, and GC letters. And then you'll notice that on the other side we have our single strand of mRNA with its complement of nucleotides A, U, C, or G. So the mRNA code is called codons, and those mRNA codes are formed from the reading of the triplet bases in the DNA. Those triplets, as we said for the DNA, ultimately will specify an amino acid. So here we have the gene within the nucleus, and then that gene is used to create a strand of mRNA, that written down recipe that's going to go to the cytoplasm to make the protein. So codons form the genetic code used to build a protein by linking together amino acids. The amino acids and the codes are shown here I don't expect my students to memorize which codes go to what. Of course, if you end up doing some genetic research and sequencing, you will eventually know these, but for clinical purposes, you don't need to memorize these codons. But I want you to notice a couple of things. First of all, notice that they're U's. So if you see a U as a nucleotide, you know that it must be RNA-based. So this is an RNA-based code. And also, look at the redundancy in the code. There are many combinations for each possible amino acid. So if we look, for example, at the amino acid phenylalanine, it can be encoded by a UUU or a UUC. That bit of redundancy is important when we think about the possibility of small mutations in the DNA, which could still lead to the same amino acid being produced. So often you'll have 2, 3, or 4 possible codons that can lead to the same amino acid being produced. So the redundancy is very important in the genetic code. All right, let's get to the goods of gene expression. So the process of making mRNA from the DNA is transcription. We're going to go in detail through transcription. And then the process of using that mRNA recipe to make a protein is translation. So take a second to write those words down. Often, because those words are very similar, students get them mixed up, but the processes are very different. We'll go through each of these 2 major processes, transcription and translation, in the next few slides. So transcription is DNA to mRNA. Translation is mRNA to protein. One way to remember it is that transcription forms the script or the recipe, and translation comes later when you translate that recipe. We'll continue with this summary figure here so you can see that we have a template strand of DNA that's used to form a single strand mRNA. And that mRNA then is used to encode an amino acid chain. This is the process of protein synthesis. So first, transcription. So transcription is the process by which mRNA is made from the DNA template. Here are the steps of transcription of DNA to mRNA. We're going to take these steps and draw them out. Again, when we're looking at the cell, this is happening in the nucleus. For transcription to take place, we're going to be taking a piece of a chromosome. So here's a chromosome. And we're going to be reading just a single gene. Of course, many genes can be transcribed at any given time, but as we're thinking about transcription, we're thinking about a particular gene being expressed. So we're really looking at one gene and how it's transcribed. So here we have a single gene on a chromosome being transcribed into mRNA. So let's look at that gene. So first of all, the gene is composed of a sequence of DNA. Remember that that DNA exists in a double-stranded form. So the first thing that has to happen for transcription is to unwind and separate the strands of DNA. So first, we have an enzyme that attaches to a region we call the promoter region. And that enzyme is RNA polymerase. the RNA polymerase attaches to the promoter region of a gene and unwinds a small segment of the DNA. So it's going to open up a segment of DNA. It'll still be wound before and after this segment of DNA. And what it's going to do then is expose the template and the coding strands. Which strand gets read? Well, what we said a couple of slides ago, it's the template strand that's going to be read to form the mRNA. So the RNA polymerase is going to separate the template and the coding strands, and then one by one add the mRNA nucleotides. So the mRNA is going to form along the template strand, one by one, nucleotide by nucleotide, forming complementary pairs with that template strand. It's going to continue until the stop codon is reached. At the end of the sequence of that gene. So the sequence of the gene includes a sequence for a promoter and a sequence for the termination of transcription. These sequences are very important for highlighting the portion of the gene that's going to be transcribed. RNA polymerase is one of the many enzymes that helps to regulate this process. I'm not going to ask you to memorize all of the different enzymes, but I want you to understand that RNA polymerase both attaches to the promoter and unwinds the DNA. And then it adds nucleotide by nucleotide to form the mRNA strand. When all of this is done, the mRNA detaches and it has exactly the complementary base pairs to the template strand. Those complementary base pairs will also exactly match the coding sequence, with the exception of switching the T's and the U's. Now that mRNA recipe is ready to be sent out to the cytoplasm to continue the process. So let's go back to our list of steps. First, we have promoter activation, where the RNA polymerase attaches to the promoter region of a gene. Then we have the unwinding, or the opening up, the separation of the template and coding strands of the DNA. Third, we have the script formation, which is forming the mRNA, adding nucleotide by nucleotide to match that complementary pair to the template strand. Finally, when the stop codon is reached, or the chain terminating region of the DNA is reached, then the mRNA will detach and the RNA polymerase will detach, break away from the DNA strand to release that final mRNA. Our recipe is ready to leave the nucleus. So mRNA is formed in the nucleus. An interesting thing to understand is that actually, technically, we would consider this to be a pre-mRNA. There's some processing that needs to happen before that mRNA is ready for prime time. So that processing includes adding a 5' cap and a poly A tail to the mRNA. It also includes some splicing or removing segments of the mRNA. So we have segments of the mRNA called exons and introns. The exons stay and the introns go. Sometimes there's a different combination of splicing that can happen, based on transcription regulation. So it's possible that from the same mRNA transcript, you can have different mature mRNAs that are formed because of alternative splicing of the introns and exons. But let's keep it simple for now and start with our pre-mRNA, or in this case, in this diagram, it's called the primary RNA transcript. It goes through processing to add that 5' cap, the poly A tail, and then do the splicing by removing the introns to get the final mature mRNA. Now that mature mRNA is ready for prime time. What happens next is the translation. So transcription of DNA to mRNA just happened, and now we're going to translate that mRNA into protein. Translation happens at a ribosome. It also requires tRNA and the amino acids, which will ultimately be bonded together to form a protein. So let's talk about those components. tRNA transfers, that's where it gets its T, amino acids one by one to the final protein. There's a tRNA for each possible amino acid and each possible codon. tRNA recognizes that triplet codon code in the mRNA. tRNA has what's called anticodons. Those anticodons will recognize the mRNA code and bring in the correct amino acid based on the reading of that code. tRNA has this interesting hairpin loop structure, or because nobody wears hairpins anymore, your textbook calls it a cloverleaf pattern. So it's a hairpin or a cloverleaf pattern to the tRNA where we have anticodons and amino acids. Ribosomes we talked about in the previous lecture on cell structure. Ribosomes are the sites of translation. This is where the mRNA docks or waits for the tRNA to come in and bring in the amino acid. Ribosomal RNA is formed in the nucleolus. It's one of the reasons the nucleolus stains so dark when we stain it with various dyes. And it's formed of two subunits, a small subunit and a large subunit. These subunits are separated until translation is activated, and that's one of the ways in which translation can be regulated, by keeping the subunits separated. So the small subunit is waiting, the mRNA docks on that small subunit, and then the large subunit comes in, forming that ribosome. Once the mRNA is docked, the tRNA will come in and bring in the amino acids. So remember that ribosomes are found on the rough ER, but can also be freely floating in the cytoplasm. Here are the steps of translation. We're going to also draw these out, but let me just tell you that there are many, many different details that we're going to glaze through for translation. There's A sites and P sites and E sites, and some of the real molecular details not necessary for us to understand the clinical application of genetic disease, but I do want you to see generally how this works. Okay, so there's a little bit of preparation that has to happen before translation can occur, and the Guyton textbook talks about the activation of the amino acid so that it can be attached to the tRNA. So remember that the tRNA has, I'm going to draw it terribly, it looks like a silly little man there. The tRNA has anticodons on one side and the amino acid on the other side. So this is first going to be activated using ATP, so this requires energy. So the amino acid is activated and then it can bind to the tRNA. The tRNA then is ready. So now we have our small subunit of ribosome, and our mRNA transcript is going to come in and dock onto that small subunit of the ribosome. It's going to start at, yep, you guessed it, the start codon. So the first amino acid that is going to always be translated is going to be that is related to the start codon. So the tRNA is going to come in and bind to the start codon. That brings in the amino acid methionine. That starts the whole process. Then the next set of codons on the mRNA will be read. A new tRNA will come in, bringing a new amino acid. Those amino acids will bond together. Let's draw that amino acid in a different color. Those amino acids will bond together, starting our peptide chain. The first tRNA will release its amino acid, and it will then leave as the next tRNA moves over to the next site. One by one, the tRNAs come in, have their amino acids bond to the previous amino acid, leave, make room for the next. The new tRNA comes in, bonds its amino acid, leaves. This process continues. I'm just going to put a ... adding one amino acid at a time, one tRNA binding and leaving at a time until we reach, guess what? The stop codon. The stop codon is going to be the last codon and it's going to end or terminate that peptide chain. So let's go back to the notes. So the preparation is activating the amino acid and getting it attached to the tRNA. Then we have initiation. This is when the mRNA attaches to the ribosome to get the process started. The first tRNA will bind to that start codon and add the methionine as that first amino acid. Then elongation, or adding amino acids one by one to the chain, is next. That's when a new tRNA comes in, brings in the next amino acid. Those amino acids bond together. This process continues one by one, elongating and continuing to add amino acids to that peptide chain until the stop codon is reached, and the stop codon signals termination of protein synthesis and the end of bringing in tRNAs and bringing in amino acids. So the stop codon is reached, the protein is done. Now that protein can go out into the cytoplasm or into the endoplasmic reticulum, depending on where it's being produced, and continue its protein function. Okay, time for a coffee break. Get your notes organized, have a good cup of coffee. We're going to come back and talk about gene regulation. For the second part of this lecture, we're going to talk about the regulation of gene expression. This is a very important concept, both for understanding cell function and also for understanding genetic disease. So the regulation of gene expression is important for how and if a protein is going to be produced. This can change the specialization of a cell. Certain cells express specific proteins while other cells do not. So within the same person with the same DNA in that nucleus, different cells will express different proteins to carry out their specialized functions. This can also be altered in order to alter cellular activity. So increasing or decreasing the amount of a protein that's produced can change the activity of cells. Finally, it can overall change the function of a cell. So the function of a cell can change based on, for example, enzyme activity, increasing or decreasing signaling molecules or enzymes or various chemical processes within a cell can alter its function. One very important understanding of gene regulation is for cell growth and cell reproduction, especially when we think about cancer cells and tumor formation, because most cancers are some misregulation of the reproduction of a cell or the cell cycle. So any step of the protein synthesis process is subject to many forms of regulation. So there is regulation at the genetic level. We can have mutations or alterations to the sequence of the DNA. There's regulation of transcription by activating or suppressing transcription of certain genes. There's activation or suppression of translation, which can change whether an mRNA is translated into a protein. And then finally, we can't forget to think about afterwards when the protein is produced. The protein can also be regulated. For example, activating or suppressing activity of a particular protein, like an enzyme, can ultimately change how that protein functions. So any level from the DNA sequence all the way to the function of the protein can create regulation of that gene expression. So first, DNA mutations. DNA mutations alter the sequence of the DNA. They can be inherited or they can happen because of exposure to mutagens. For example, UV light or a chemical mutagen exposure during the course of our lifetimes. Here I'm showing a mutation caused by UV light, where you can see alterations in the DNA structure have happened because of exposure to UV light. If there's a single base change in the DNA, that's called a point mutation. So here in this diagram, you can see some examples. So look down here at the nucleotides. Here at the DNA level, the sequence or the triplet was a TTC. And here are all the possible mutations. So there could be no mutation. There could be a simple change, and for example, a TTC to a TTT, but that still encodes for lysine, the amino acid lysine. So mechanistically, there's no change there. So that's a silent mutation. It didn't matter. The protein was still identical. A nonsense mutation is a mutation that changed from a TTC to an ATC, and that ended up forming a stop codon, and now there's nothing to be read. A TCC mutation, a single change in the T to C, caused the change to a different amino acid. And that means that it's a missense mutation. A missense mutation changes the actual amino acid and the protein structure. In other words, it's a mistake in the protein structure. There are also chromosomal level alterations in sequence. These are larger segments of genes that can be either deleted, duplicated, inverted, translocated from one chromosome to another, many possible chromosomal changes that can alter the sequence of DNA. When we talk about the cell cycle and meiosis, the reasons for these chromosomal alterations will make more sense. So more next week. In addition to alterations of the DNA sequence, there is also regulation of transcription. This is a very important way in which gene expression is regulated. And you're going to hear the term transcription factor quite a lot. So transcription factors are factors or molecules that can change the transcription of a gene. Transcription factors can be enhancers, meaning they can increase the expression of a gene, or repressors, meaning they can prevent or decrease the expression of a gene. There can also be other alterations that change transcription. For example, changes in the promoter region. Once the mRNA is made, transcription is done, there can now be translation modifications or alterations in the translation of that mRNA to the protein. These are less common than transcription alterations, but they still can happen. So there can be initiation factors and regulatory factors that can affect translation. Another way that we already talked about is actually the splicing of the pre-RNA to the mRNA. That's a common way in which translation is modified. You can have alternative splicing or regulation of the splicing of the mRNA. In addition, in recent years, the research has shown the presence of microRNAs, tiny, small segments of RNA, about 20 base pairs, that are complementary to the mRNA within the cell. These microRNAs can do a lot. MicroRNAs can affect transcription and translation. So microRNAs, for example, can bind to the RNA, bind to the mRNA and lead to its degradation, and then that mRNA will never be translated. So microRNAs are a pretty interesting area of research, probably getting to be about the last 7-8 years that research has become really cool. And lastly, protein regulation. So as I said a few slides ago, we can't forget that the protein itself can be modified. The structure of the protein and the protein folding can modify its function. The actual protein can be degraded. Remember proteasome and other degradation mechanisms within the cell. And then finally, the activity of, for example, enzymes can be inhibited or activated by various cofactors in the expression of other proteins. So again, gene expression can be regulated all the way from sequencing to translation. And don't forget, all is not lost. You can still modify the protein once it's made. All right, that's it for genetics. I hope this was helpful. If you need more, please see Guyton Chapter 3. One note, we didn't cover cell cycle today. That's at the end of Chapter 3. We're going to cover that in a separate lecture. If you'd like to do some challenge questions, stick around. I've got 2 really good ones for you. See you in a bit.